Welcome to MedTech Global Insights. Today we are demystifying a critical standard for medical device safety, ISO 10993-1, which governs the biological evaluation of medical devices. This isn't just a regulatory hurdle, it is the cornerstone for ensuring a device is safe for human use. The most significant evolution in this standard, particularly in its 2018 revision, is the definitive shift from a simple checklist of tests to a comprehensive risk management process. This means you can no longer just tick boxes. You must think like a detective. Your biological evaluation must now be driven by a thorough risk assessment of your device from start to finish. The process begins with understanding your device intimately. What materials is it made from, including any colorants or additives? How is it manufactured? And could any processing aids leave residues? Most importantly, what is its intended use? This is defined by the nature and duration of body contact. A surface electrode with short-term skin contact has a vastly different risk profile than a permanent cardiovascular implant. For the electrode, tests for irritation and sensitization might be sufficient, but for the implant, which has long-term contact with circulating blood and tissue, the evaluation becomes far more complex. Let's consider a real-world example: a new drug-eluting coronary stent. The device itself is a metal alloy. It's coated with a polymer, and that polymer releases a pharmaceutical agent over time. The risk assessment must consider all three components. You need to evaluate the biocompatibility of the base metal. You must assess the polymer coating for any potential toxic leachables. And you have to understand the biological effects of the drug itself. This requires a detailed biological evaluation plan, or BEP. This plan documents your entire risk analysis and justifies your testing strategy. It might conclude that you need to perform tests for genotoxicity to see if any leachables could damage cellular DNA. It would certainly require hemocompatibility testing to ensure the device doesn't cause dangerous blood clots, and it would necessitate long-term implantation studies to observe tissue response over months or even years. This risk-based approach also emphasizes chemical characterization, as detailed in ISO 10993-18. The goal is to identify and quantify the chemical substances that could be released from the device during its use. A robust chemical characterization can sometimes be used to justify not performing certain biological tests, potentially reducing the need for animal testing and saving significant time and cost. Failing to conduct this comprehensive, risk-based evaluation is a common reason for rejection by regulatory bodies like the FDA or European notified bodies. It results in costly delays and additional unplanned testing. Navigating these requirements demands deep expertise, a task that can be daunting for even the most innovative companies. For guidance on creating a robust biological evaluation strategy for your medical device, you can email us or visit our website. Contact Pure Global at info@pureglobal.com or visit us at https://pureglobal.com.